Everyone, good morning, namaskara. A warm welcome to everyone to the second edition of the HDSI International Conference here at NIMHANS. We are delighted to have all of you with us for an incredible gathering of scientists, clinicians, caregivers, people with lived experience, and advocates from around the world. Over the next 2 days, we will explore the future of HD research and advocacy in India, and I am happy to welcome all of you today. We will first start with a technical talk, and for that I am happy to introduce the chair of the session, Dr. Biju Viswanath. He's a clinician, scientist and psychiatrist at NIMHANS. He's in charge of a molecular genetics lab and CBM, Center for Brain and Mind, where we have been looking to understand the molecular biology of neuropsychiatric conditions.
Good morning. So it's my pleasure to invite the first speaker for the day, Dr. Subramaniam. He is an associate professor in the Department of Chemistry and Biochemistry at Florida Atlantic University. He earned his PhD in Natural Sciences from the University of Heidelberg, Germany, and completed postdoctoral training in the Johns Hopkins University School of Medicine. His research focuses on the molecular mechanisms underlying neurodegeneration with an emphasis on brain-enriched proteins that regulate movement, cognition, and cell survival. His lab is known for pioneering work on the intercellular spread of mutant Huntington in Huntington's disease. He has received numerous honors, including the Alzheimer's Association International Research Award and the Daniel Nathans Research Award, and was invited young scientist at the Lindo Nobel Laureate meeting. With that, I invite you, sir, to give the talk.
Okay. Thank you, Vijay, for a nice introduction. I want to thank organizers for inviting me, particularly Meera, for this great opportunity to be here. When I started HD, told me 20 years, HD research 20 years ago. You can't hear? Can you hear now? Okay, great. Thank you. So when I started Huntington disease research 20 years ago, it was always thought as a disease of Caucasians origin because there's not much data available in other part of the country, particularly in India. So I want to thank HDSI now to really make this a important event to bring awareness, particularly NIMHANS for really making this as an important step in understanding disease, particularly neurodegeneration. So this meeting, I think, probably going to happen every year now onwards, and there will be more awareness and more people coming together to resolve this disease. So today, I just want to present some of the work we have been doing in our laboratory. Particularly, it is on understanding how the disease potentially spread from one part of the brain to another part of the brain. So I want to begin with this particular slide. You can see here, these are the elite runners. They compete in big marathon events, and you can see all these 5 elite runners, they exchange water just using one bottle, So it's a physical transmission of this important element of life, the water, and then they kind of throw it off. So the idea is this interaction is so crucial for human survival. I know interaction can be bad and which results in other issues. So the reason I showed is that such a physical interactions are critical, and probably the cells in our body, they constantly interact with each other. And those interactions keeps us actually who we are, how the organisms functions. So apparently, as you all know, we are gathering here to understand the disease mechanisms and find therapies. The Huntington disease is a motor disorder, and it affects the fundamental functions of the body, ability to walk, ability to sit, and this is a major problem, and we are here to really understand and go forward on this disease. So the problem the good news about Huntington disease is that we all know the gene that is involved in the disease process. So it's a Huntington gene called Huntington. The problem is in the N-terminal part of the Huntington, which normally codes for around 6 to 35 CAG repeats, and that's considered normal. And if you have anything more than 35, then you have a tendency to get Huntington disease. And if you're really many number of repeats, CAG repeats, then you're going to have an early onset. It's a juvenile Huntington disease. So the difference between normal and Huntington disease is only the CAG repeats, which codes for glutamine. So that's the only difference as far as we know. So then the question is how this gene impacts Huntington disease patients so profoundly. So here is a study done by many groups across the world, and these are the MRI images of Huntington disease patients. As you can see, the clear loss of striatum. This is the region close to the lateral ventricle. That's the striatal region. You can see in Huntington disease patient, that region is almost gone compared to the normal healthy subjects. And what is well known in the field is that the disease begins in the striatum and then slowly spreads to the cortex. The cortex is another region impacted significantly. For example, regions such as cerebellums are not so much impacted. There are not so great significant symptoms related to cerebellum. This is kind of well accepted idea that the Huntington disease, the striatum is the most vulnerable region, and then it spreads to the cortex. And this particular slide, I like it. This is a longitudinal MRI of Huntington disease patients. What you can see here, this is the data is taken for 4 years, 2008 to 2012, of the same patient. That is the striatum in 2008, where my pointer is. Within 4 years, there is a significant shrinkage of the striatum. So that's a big change. And also you see some changes in the cortex, but not much on the cerebellum per se. As you all know, the striatum is important for many of these functions, as mentioned here. Motor action, motor function, decision-making, motivation, reinforcement, reward. Almost all important functions are regulated by striatum. And the striatum is degenerating, and the patient have this experience of not performing in the fundamental functions of the brain. So the major question is the mutant Huntington that I mentioned. Well, there is expansion of CAG repeats, and that protein is present everywhere in the brain, even in the peripheral tissue, more or less equally distributed. But why does it affect striatum? What is so special that the striatum is vulnerable is the first part of the brain which is impacted by the disease? This is a question that still we are not able to fully understand. So today in my presentation, I wanted to bring some perspective on this based on the work we have been doing for many years now. So here is the protein called RAS, which is a RAS homolog enriched in the striatum. As the name says, this protein is highly present in the striatal region. This is the mouse brain, and here is the protein expected size, and is also modified in the brain, the striatum. The protein is present on the plasma membrane. Actually, it binds to the membrane, and it's heavily modified by another protein called SUMO. And the protein also has a E3-like domain. That means it can simulate other proteins. It can modify other proteins. And this is the modification, as you see here. The protein itself is modified. And the distribution of RAS in the human brain is also highly enriched in the striatum. This is what's shown here. So, and the protein is present mostly in the medium spiny neuron. This is one set of neurons, highly abundant neurons in the striatum, which are vulnerable, which are degenerated. And this protein is present in those neurons at high concentration. So what we have found is that this protein has some unique property. As I mentioned, it can really interact with other enzymes which can modify the proteins, and those interaction it can really changes the biochemical properties of the protein. So they exist in the complex sumo complexes as it's shown here, and when they interact with the target, which could be Huntington, I'm going to come back to that. So it's going to change the nature of the protein, and that could be having problem. So over the years, what we have found how this protein may be causing the problem is that Res protein in the striatum interacts with Huntington, particularly the mutant Huntington, the expanded version. It has more affinity toward that protein and modifies it by adding another small protein, which is SUMO. And when that happens, the protein becomes more soluble. That means more available to cause damage. So that soluble protein now can interact with other cellular machineries, for example, regulating mTOR pathway, one of the important pathway for aging-related mechanisms, or also it can interact with the ribosomes and affects the translation, which is also a very fundamental function. When these things happens in the striatum, because of Res, then the striatum starts having problem. This is our kind of working model. So I'm going to throw some data. These data is already published, as you see, long time ago. But what we found is that if you take a mouse model, there are mouse models available for Huntington disease-like symptoms. And then you remove Res from those mice. And you can see, you can protect those mice. For example, here, what you're looking at is some of the common tests used for Huntington disease motor assay. Like Huntington disease mouse model, they have a reproducible motor problem, just like what you see in human patients. Here is Huntington disease mice not able to stay on a rotating rod. Normally the mice learn to stay on the rotating rod, but here this Huntington disease mice cannot stay. Whereas the normal mice, they can keep doing better over time. But if you have Huntington disease mice without Res, either one copy or two copy, you can see they have some protection, clear protection from the motor deficit. Here we did many other tests. You can see the ledging, clasping gait, kyphosis and tremor. In Huntington disease mice, they really behave bad. When you remove Res from those mice, and those mice seems to do better. And here is also we looked at the anatomical change. Anatomical change is not the biggest, the clear indication of the disease progression. This is Huntington disease mice. You can see it's the lateral ventricle, which is an indirect measurement of how big is the striatum. You can see that's really big in Huntington disease mice, which is kind of quantified here. When you remove 2 copy or 1 copy of Res from those mice, they kind of reverse back to those problems. So the deletion of Res is really protective, and this they look like more like a wild type striatum, the normal mice. Then we had another question. Okay, if this protein is so important for causing Huntington disease, then what happens if you express this protein in the cerebellum of the mice? So if it is so important, it should have some consequences of that. So here is the Huntington disease mice. At this point, they do not have any symptom. They kind of walk around normally. They kind of get up and stretch nicely. So this is how the mice normally behave in an open environment. So here we did a surgery. This one, the mice, they just have a control injection without any Res, and they can see they kind of walk around and then they walk away. But then if you, for the same age group of mice, Huntington disease mice, if you inject Res in this mice, see this mice within few weeks, and they starts having some kind of problems, which is obvious. You can see they're not able to stand. They seems to have kind of ataxia-like feature. So this again suggests that this protein in presence of Huntington is actually toxic to the mice. So then, if you look at pathology of this, if you look at the cerebellum of these mice, and you clearly see that one of the major neurons in the cerebellum, they're called parvalbumin neurons, and they kind of almost, we don't see any expression of that protein. We believe that they are kind of dead. And then this is you see how much loss of the paravalvular neurons in the cerebellum because of rest. And then more importantly, as I mentioned in the beginning, that the rest actually increases the soluble form of Huntington. Because that is the control mice. Then you have rest and Huntington. You can see there's a large increase in the soluble form of Huntington. Then they also increase in caspase-like activity, suggesting that the cells are dying. So this is kind of all coming back to the idea, the model, that why does striatum degenerate in Huntington disease is probably because of this interaction of rest and Huntington and doing all this damage that I described. So this model is not only we kind of identified, of course we did the first identification, but later many other groups independently verified this model using very different kind of cell model, mouse models, human iPSC. They kind of come up with the similar idea that rest could be toxic in Huntington disease. And this model works. So up to this, everything went fine, but something happened. Something unexpected happened. So what happened is that the protein itself, the rest protein I described, it itself can travel from one cell to another cell. It's going to physically travel. That process, using a process called tunneling nanotube. So what are tunneling nanotubes? So these are all the structures which are made between the cells. The dynamic structure, most of the time in cell culture, they're like straight road made up of membranes, and they can transmit proteins. It could be pathological proteins, it could be prions, it could be HIV. So this protein, TNT, act like a conduit for transmitting cargo from one cell to another. As you can see, this is a very newly emerging field. You can see a lot of paper coming up. I should update this, but you can see this is a growing field that many people are looking at this structure. But are the TNT-like structure exist in the brain is not fully clear, but a lot of evidence slowly coming up now. These are the structures which are important for neuronal functions. So again, this stage we are at a very preliminary stage on this. So what happened with rest? So with rest, you can see here, we express rest in any cell type. This is a striatal neuronal cells. You can see it starts making these connections. And these structures are similar, described in Drosophila cytonyms, like a TNT in PC12 cells. And if you do a scanning electron microscopy, you can see this is a seamless transition of this tube from one cell to another cell. And they're membranous, and they also consist of actin. So how many number of TNTs can be formed? Again, if you have a cell which has no rest, for example, in this case, you can see it can make many connections between the cells from one cell to another. They're very dynamic, they're actually formed. And also importantly, whether rest can make TNT in its natural environment, which is a medium spiny neuron, which is affected in Huntington disease. You can see here, normally this is how the neurons look, nice processes, cell body, etc. But when you express rest in those neurons, you can see these neurons now forming a long tunneling like nanostructures coming from cell body or the processes. And this is kind of interesting, like this is really a new field. Here I wanted to show you how these structures can transmit cargo. So in this case, what you're looking at is basically a co-culture. One cell is colored with rest, another cell is colored with membrane marker. You can see here there are you saw two TNTs. One of the TNTs delivering the cargo, and after some time the cargo enters into the cell. So this is going to be replayed again. There are two TNTs, highly dynamic. There's a lot of tension there. One of the TNT delivers the cargo. You can see it's delivering the cargo, and this cargo travels along the membrane here, and then after some time it's getting internalized. So it is actively making these tubes and actively transferring, getting into other cell. So what does this do in other cell? We still don't know, but we wanted to know whether these TNTs can transmit Huntington, because that's our main question, right? How does it relevant in terms of Huntington disease? So what you are hearing is one TNT here from this cell, and that TNT is positive for Huntington. You can see these punctures, and here is kind of expanded one. Inside you can see the zoomed version. The TNT obviously contains Huntington, so Huntington seems to be getting from cell to cell. In this video, you also can see that the TNT is now this is a puncta of Huntington and rest together, and it is getting into the other, trying to getting into other cell. Most of the time, each puncta of Huntington, which is traveling in TNT, also positive for rest. So this seems to be co-traveling most of the time. So Huntington itself is not traveling much. So it requires rest as a vehicle to move it from one cell to another cell. Here's another video I wanted to show you. You can see here is the rest and Huntington. This is a TNT coming from somewhere else. It's from some other cells it is coming. You can see it is now transferred a cargo into, already transferred the cargo, one cargo, and the second cargo is getting transmitted. So it seems to be like very, like it seems to pierce the other cell and deliver the cargo, So that is interesting. So this is a quantification data, like we want to know how much is going and whether it is Huntington, wild type Huntington. So we did all this study. Basically, as I mentioned, the Huntington itself is not transmitted much. The wild type Huntington is not at all transmitted, very little. But in presence of rest, the mutant Huntington is really transmitted extensively between the cells. So that is interesting. Then again, this study is done in cell model, but we wanted to ask the question whether this kind of transmission, Huntington transmission, can happen in real brain. So I should mention that it's already known in human patient studies where they do some transplantation work. They saw that the mutant Huntington is entering into the normal grafted tissue. They're surprised to see mutant Huntington in a normal grafted tissue, in a postmortem tissue. So it's happening. Huntington is moving from one cell to another cell. They just showed it in Drosophila model, where you express Huntington in olfactory bulb, then the Huntington is spread to other part of the brain, and in mouse models, so on and so forth. But we still don't know the mechanism how Huntington is transmitted like this. So we wanted to test in our model whether this kind of transmission can happen in vivo. So for that, we needed a tool. We needed to know where exactly we are putting our Huntington. Then we can track them, right? So for that, we use this tool, which is commonly used. We make one mRNA. We can make two proteins. So this mRNA, through lentivirus, we express them in wild-type brain striatum and also rest knockout striatum. So we use both normal and wild-type Huntington just to do a good control studies. This is the expression pattern of normal Huntington and mutant Huntington. So the expression is fine. We wanted to know when you express these one mRNA in these cells, how these two proteins are spreading. So if Huntington itself is spreading, you should see only one color, right? Because this is coming from one mRNA. If both proteins are spreading, you should going to see two colors. So here is the injected spot. This is the striatum of wild-type mice where we injected lentiviruses having a mutant Huntington and just a reporter. Because coming from one mRNA, every cell is red and green here. But if you go further down, 500 micron away from the injected spot, and just zoom that part here, you see only red spot, which is Huntington. So that means Huntington from here is moving away from the injected spot, because we can clearly see that a lot of perinuclear accumulation of Huntington in this spot. Then if you do the same experiment in the rest knockout mice, you can see this is injected spot here. But if you go further, you see very few punctures compared to the wild type mice. This suggests that you need to have a rest to really propel this movement from one place to another place. And if you quantify here is the injected spot, you see there's a clear decreased transmission of Huntington Normal Huntington is not transmitted so much. And in the injected spot, away from the injected spot, you can see a dramatic decrease of transmission of Huntington. So this confirms that whatever we are seeing in vitro, the transmission happening between the cells, so RES is also doing it in vivo. I will take few more minutes to really mention, like, okay, this seems to be interesting, but what are the mechanisms? Why would this RES makes this a road? Why would it, and how does it? Why would, we don't know yet, but how? How does it make these tunneling nanotubes? So we wanted to address that question. So we took use of this information where RES has to be on the membrane. So this mutant of RES, which is called cysteine, which is a farnesylation mutant. So farnesylation site, that site is required for binding it to the membrane. That means if it binds to the membrane, it can make this structure. If it is not binding to the membrane, it cannot make structures. Then we wanted to, that is the case, we wanted to ask this question, okay, it binds here and makes a TNT, and if it is not, then no TNT. Then we want to know what is that it is interacting on this cell membrane. Particularly we're looking at protein interactors. We believe that those kind of protein machineries are involved in this. So with this approach, again, this is just showing you what we did. We used all kind of controls here to really isolated the membrane. Then we co-immunoprecipitated, isolated RES from the membrane, and we did mass spectrometry to really look at all the components, all the protein components are binding to the membranous RES. As you can see, a lot of non-specific binding, but we were focusing on the one that is only present in the wild type RES. So with this approach, we identified around 200 proteins. Here is the top 10 proteins that is listed here, and the top one is all the time we saw one of these is called transporter called SLC4A7. I never heard of this protein before, but it's a transporter involved in the pH regulation. I'll come back to that in a minute. And we also saw other like plastine-2 and so on, the many different proteins. Then we did a screening, unbiased screening, to see which one is reducing the TNT formation. As you can see, SLC4A7 dramatically decreased, significantly decreased, and followed by plastine-2. Plastine-2 is actin-related protein. We knew from previous studies, that actin is important for TNT formation. So this is not surprising. But what we were interested was SLC4A7. So SLC4A7 is a transporter. So this is basically a pH regulator. It transmits sodium and bicarbonate. I'm seeing a cough myself there. Okay. So you can clearly see that this protein has got around 14 transmembrane structures, and basically previous research showed that it is involved in hearing and locomotor behavior, and also if you remove SLC4A7 in the mouse model, it can prevent NMDA-related seizure and neurotoxicity. So there is some connection of this protein already with a toxic property, and most studies clearly demonstrate that it's required for maintaining intracellular pH. So how does the pH is involved in TNT formation is still an open question, but looks like this property is important. So what we did is we just deleted SLC4A7. You can see this is a lot of TNTs in this structure. When you delete SLC4A7, deplete, you see less number of TNT formation. Interestingly, luckily, we had also an inhibitor of SLC4A7. They're not a specific inhibitor, but there's it binds a family of SLC4A protein. There are 10 members in this group. If you use that inhibitor, also you can decrease number of TNT formation. So clearly, whether it's a genetic model or a pharmacological model, you can block the TNT formation when you block the SLC4A7. We were interested in looking at whether there is the localization of transporter in the TNT. You can clearly see here is the RES coming from this cell. There's a bunch of TNTs here, and they're also positive for SLC4A7. And here is the more high resolution imaging, STED imaging, and this is the TNT consists of both SLC4A7 and RES. And as you can see, they're very closely associated, even in the TNT. This just shows you how close they are in the TNT. So they are interacting. They are also like physically interacting. From the study, we evaluated the physical interaction using many different methods. I don't want to go into the detail of that. Basically, this shows that these proteins have the high chances of interacting directly each other. They're interacting directly between them. So here is the chimera model showing that how these proteins could be interacting, and this is SLC4S7, and there's a RES here, and this sumo component, as I mentioned, RES is important for simulation. It also interacts with other component of simulation. The whole complex seems to be high chances of interacting each other and regulating TNT formation. As I mentioned, like this, one of the properties of SLC4S7 is regulating pH, and what we found is that if you use the reporter, the pH reporters, you can clearly demonstrate that if you inhibit the SLC4S7, you can decrease pH, and if you have RES, it can increase pH. But then if you again block SLC4S7, you can decrease the pH activities in the cell, suggesting that RES and SLC4S7 interaction, the binding not only we know that it regulates TNT, but those interactions seems to also regulate pH somehow, and that may be linked to TNT formation. Finally, because our intention is to look at how this interaction regulates Huntington transmission, our goal is to go behind these complexes and maybe inhibit them. So here what we're looking at is that if you block SLC4S7, you also block Huntington transmission. So that's kind of further establishing the fact that we now have two targets to target. Either you can target RES, which is relatively harder, but you have SLC4S7, which is a transporter. A lot of studies are coming up showing that you can use antibody against SLC4S7. So that gives an opportunity like, you can really block Huntington transmission and see whether they have any therapeutic effect. So we also looked at the brain of this SLC4S7 knockout. So we got this mouse model from a collaborator in Sweden. So we did the same thing. What we did for RES work, we injected Huntington into the striatum, and then we followed up, and we can clearly see that here is the quantification that the influence of SLC4S7 knockout have a diminished transmission of Huntington, whereas the other control signals are not affected. Again, this further strongly establishes the role for SLC4S7 as a regulator of Huntington transmission in vivo in the striatum. So just to summarize this model, how this TNT formation may be happening, so we believe that on the membrane, RES interacts with SLC4S7, and through this carbonate sodium transport, and they regulate pH, and this pH may now impact actin bundling, which is well known that pH regulates actin bundling, and through those the molecular changes, now the TNTs start forming. Again, what is the signal for TNT formation? We still don't know, but we believe our data support this model of formation of TNT. Again, finally, what does this all means to in terms of disease per se, right? I begin with the slide showing that the disease. Now most people accept the idea that disease starts in the striatum and spreads to the cortex, right? And how does this happen? How our model, TNT model, can be used to really understand this? So what we believe happening is in the striatum, there is a collateral connections between medium spiny neuron, and RES and SLC4S7 transmit mural Huntington, and that transmission may create a homeostasis problem. It may block the regular transmission, physiological transmission perhaps, and that may impact striatum. And then Huntington now also can be transmitted from the striatum to the cortical projection. There's a lot of projections from the cortex get into the striatum, and there is this transmission may be happening from the striatum to the cortex. And when these processes are happening, now the striatum is impacted, and slowly the cortex is impacted. So this is the model that we are currently working on. So I wanted to thank all the people involved here. As you see here, many people involved in this project, and we are very excited. Really continue to work on this project, and I also want to thank my funding agencies, and of course, without them, I wouldn't be here. Thank you for your attention. I'll be happy to take any questions that you may have.
Thank you for the wonderful talk. I'll open the house for questions.
Yes.
Yep.
Hi. Yeah. Hi. Really great talk. I was wondering if the repeat expansion that is present on the mutant Huntington, how does it affect the interaction of RES and mutant Huntington protein?
It's a very good point. So we did the kind of screening where does REST bind. So it seems to be that this huge glutamine expansion seems to give additional affinity, and that seems to be the main reason for a stronger binding. But how does it actually are regulated? We don't know, because we don't have a structure for that. We don't know how it is really interacting and all. So the only thing we know is the expansion somehow offers the extra affinity to RES.
Because I was wondering that would be a really great pharmacological target to then help with the
I agree
disease.
with you. I agree with you. I know several people, even in NCBS, we have collaboration to really look at the structures and then really go behind that. You know, the REST protein is very sticky protein. It's really hard to purify that. But yeah, we are on the process of getting there. It's activity, but otherwise they're not lethal. They reproduce. They're kind of This seems to live normal. Yeah.
Hi Srini, very
Hi.
nice talk.
Thank you.
My question is, how important might it be for these pathogenic versions of the protein to move? Because after all, the mutated protein is present in almost every cell type, probably also in cortical neurons.
Yeah.
So what fraction of the pathogenicity is due to the movement itself?
Yeah. So this is a very good point. One reason that I can answer the first question relatively easy is also provided by data. One reason that maybe Huntington is there everywhere, but in terms of the way its biochemical nature may be different.
Sure.
Like in RES, in striatum, maybe it's present in a highly soluble form compared to other part. Maybe that soluble, maybe oligomeric soluble form may be more toxic. Maybe cells may be now trying to get rid of that. But then the whole process turn into something else during the process. Although it's begin as a protective mechanism, but it ends up having some other issue. That's what my answer to the first question. But the second question, what is the second question?
I was wondering if there isn't already enough of those proteins, but I think these two are related. They're related.
You're right.
So
Yeah.
you're thinking that along with HTT, this whole complex of things are going which can make the intracellular milieu more toxic because
Yeah.
of movement.
Exactly. But you are asking a very important question, like how much it is movement related. We don't have any answer. It will take several years to answer that question.
Sure.
Because we need to have machineries or tools to really block those transmission in a way. And one thing we're doing is we wanted to express Huntington in one location and then see whether during the spreading, when this Huntington spread from cell to cell, there any cellular phenotype is generated, like some caspase-3 or some glial response or something like that. That is in the
Yeah, I was going to ask about glia.
Yeah, exactly. Yeah.
We'll talk later. Thank you.
Thank you.
More questions? Yep. Behind.
Yeah, a great talk, Srini. So one question already been asked by Shiva. So second is that SLC4A7 or RAHS, they have normal functions also. So when you knock out or inhibit them, they will have some side effects. And further, that in the wild type expressing cells, why don't RES or SLC4A7 do anything, or there is no TNT form. So what is the exact reason that TNTs are formed only in the mutant expressing cells, not so much in the wild type expressing cells?
Yeah, actually, TNTs are formed irrespective of Huntington. So when the Huntington is there, it can be now transported through the TNT. So the formation of TNT has nothing to do with Huntington.
You in one of the experiments you showed, it was preferentially transporting Huntington, mutant
Yeah.
Huntington, but not the
Yeah,
GFP.
You're right. That happens because the RES interacts strongly with mutant Huntington, and then it co-transmits on the TNT. So if there is no mutant Huntington, the TNTs are still there. The other cargos are transmitted. Endosomes are transmitted. Lysosomes are transmitted. And what they're doing on other acceptor cell, we don't know yet. But when there's a mutant Huntington, now it can be efficiently transmitted through the TNT. Yeah.
Thanks.
Thank you.
Yep, on this side.
Oh, sure.
It was nice talk, Sini. Yeah.
Nice to-
My question is, when you express the wild type and the mutant, whatever the pathogenic version of the Huntington within the cell, how the distribution happens when you do with the lentivirus? They are localized in case of the pathogenic variant of Huntington, or it's kind of evenly spread out within the cell compared to the wild type?
See, depending on the construct that we use, if you have a fragment of Huntington and it forms a lot of aggregates.
Within the cell.
Within the cell,
Okay.
yeah. And if you use a full-length Huntington, the propensity to form aggregates are very less. And you won't see the aggregated form, more amorphous distribution within the cell. Yeah.
Right. So you both wild type and the pathogenic variant, they are kind of distributed. There is a full length.
Yeah.
Okay.
So you can clearly see they're different. Even in fact, the full-length Huntington, mutant Huntington, people believe it doesn't even form aggregate or visible aggregates. Yeah.
Okay.
Yeah.
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 Thank you.
Okay. Imtiaz?
Hi, it was a nice talk.
Thank
So actually, in one of your videos, you showed that means the movement of the wild type mouse, and when you injected RES protein, I mean the mouse showed the Huntington phenotype. So I'm wondering that because you mentioned that the presence of mutant Huntington and RES both are required for the phenotype of Huntington. So how? After only injecting RES, it started showing the phenotype of Huntington, where mutant Huntington was not present.
Yeah. Don't have to slow. So if you express RES alone in wild type mice, nothing happens. If you express RES in the cerebellum of Huntington disease mice, because of Huntington RES interaction, now the Huntington start changing its biochemical property. And that is why it becomes toxic. Cerebellum becomes toxic because of the RES expression.
Mm-hmm.
So RES in the wild type do not cause any problem.
Okay.
Yeah.
And the second question is that
I think we will stop. You can take it up in the session.
You can talk when you meet.
Yes, I think.
Thank you for the questions.
With that, we will end this session. I thank Srini for a wonderful talk, and thank you all. Let's move on with the next part of the program.